The overall goal of this procedure is to illustrate an easy-to-use ex vivo model to investigate fresh carotid or coronary artery plaques. This is accomplished by obtaining fresh plaques from patients undergoing endarterectomy or coronary artery bypass. The second step is to cut the specimen into small pieces. Next is the random distribution of plaque pieces into a 48-well plate containing RPMI medium. The final step is adding a substance of interest, such as cytokines or chemokines, alone or in combination for defined periods of time. Ultimately, RNA isolation, qPCR, immunohistochemistry, and ELISA can be used to analyze the plaque culture experiment.
The method introduced here can help answer key questions in the vascular inflammation field, such as increasing our knowledge on the pathophysiology of human atherosclerosis. The implications of this technique extend toward diagnosis and therapy of atherosclerosis, because this is the only method to study the role of interesting molecules on the inflammatory milieu inside human atherosclerotic lesions. By this, it may allow the identification of novel therapeutic targets and development of novel therapies. Demonstrating the procedure will be done by Nadine Bamskans, an excellent technician from my laboratory.
Begin this procedure by adding RPMI medium to a cell culture dish. Place the plaque tissue in the dish such that it is completely covered with medium. Then,